Lipids, an organic molecule shaping every cell since the dawn of life 3.5 billion years ago. With applications relevant to drug delivery in medicine, to cleansing properties in commercial detergents, and flavor in food. Inquiry into these molecules has become a hotbed for scientific investigation in recent decades. Lipid-based discoveries have already begun to change our daily lives and may soon promote our species' longevity. Take a journey with me to discover life on our plasma membrane. Our understanding of cell membranes has not always been as sophisticated as it is today. Originally, scientists believed that the cell membrane was a fixed structure with cell membrane compositions differing from cell to cell, each with a unique architecture. It wasn't until 1972 when two bold researchers challenged the status quo. S.J. Singer and G.L. Nicholson proposed a new paradigm to describe the cell membrane known as the fluid mosaic model. The classical model described the cell membrane as a trilamellar composition with a lipid bilayer between two protein monolayers. Singer and Nicholson argued that the model did not maximize hydrophobic and hydrophilic interactions of the membrane phospholipids. In contrast, the fluid mosaic model describes a protein-packed membrane with greater fluidity in which the proteins sit in a lipid matrix. Before this paper, scientists believed membrane diversity was too vast to generalize in any useful way. After the fluid mosaic model, scientists can better describe the membrane on a thermodynamic and systemic level. Although controversial at the time, the model described by this paper has become fundamental knowledge now taught at the high school level and has accelerated scientific advancement into cellular biology. From these discoveries, scientific inquiry has progressed tremendously toward topics that would have otherwise been unapproachable, one of which is the role of membrane curvature by lipids. Lipids may possess a negative, neutral, or positive curvature. Lipids with a negative curvature such as phosphatidylethanolamine, diacylglycerol, and phosphatidic acid have a cone shape and produce a concave membrane. Lipids without curvature such as phosphatidylcholine, phosphatidylserine, and phosphatidylinositol have a cylindrical shape and produce a flat membrane. Lipids with a positive curvature such as lysophosfatidylcholine have an inverted cone shape and produce a convex membrane. The stability of the membrane is determined by the composition of all three of these lipid types. Cone-shaped lipids produce curvature stress into the membrane, causing the phase transition temperature to decrease, and the lipids to behave in a disordered fluid phase. It can be stabilized by incorporating cylindrical and cone-shaped lipids. Stabilizing the membrane means that the phase transition temperature increases and the lipids behave in a more gel-like phase at higher temperatures. Phospholipids with negative curvature allow for efficient import and export of transport containers by reducing the energy required to adapt. For instance, the conversion of LPA into PA by endophylline bar domain proteins allows the cell to quickly produce negative curvature into the membrane. This is particularly important during fusion and fission events, which may otherwise be energetically straining. Understanding the fundamental properties of lipids and their role in the cell membrane has led researchers to create man-made transport containers called lipid nanoparticles. Lipid nanoparticles are merely vessels designed to carry valuable materials into targeted cells. They have the potential to treat a multitude of ailments depending on the drug they are loaded with. The difficult part about administering LNPs is that the body's immune system has evolved to detect and destroy foreign particles. To resolve this issue, LNPs are coated with polyethylene glycol or PEG lipids to mask them from the innate immune system. The PEG lipids act as a polymer brush that blocks opsonins from coating the LNP surface and promoting their adsorption by macrophages. PEG lipids' negative charge also masks any cationic charges, which would otherwise cause them to be cleared by the immune system. The higher charge density also prevents LNPs from aggregating together. Lastly, longer acyl chains on the PEG lipids increase the circulation half-life of LNPs, as shorter acyl chains tend to dissociate from the LNP faster. To deliver LNPs to a targeted tissue, researchers may incorporate specific ligands into the LNP's outer leaflet. To target liver tissue, for instance, endogenous apolipoprotein E, ApoE, is pre-incubated with LNPs to facilitate adsorption by hepatocytes, similar to the native adsorption process. Tumors are also a popular target for LNPs due to the enhanced permeability and retention, or EPR, effect. The EPR effect takes advantage of the large fenestrations between the endothelial cells and poor lymphatic drainage of tumor tissues due to their rapid angiogenesis. This allows LNPs to aggregate in tumor tissues and release their contents with greater precision. Once the LNP is taken up by the endosome of the target cell, a secret ability of the lipid activates. You see, researchers optimize the lipids used in their LNPs, particularly their ionizable cationic lipids. As the endosome becomes more acidic, the ionizable lipids are protonated and take on a positive charge. These cationic lipids then associate with the anionic lipids of the endosome to form destabilizing structures. This disrupts the endosome and releases the contents of the LNPs into the cytosol. Although this form of drug delivery is very promising for treating various diseases, it is not without its shortcomings. Look at plasmid DNA delivery by LNPs, for instance. Even if a researcher successfully delivers the LNP payload to the cytosol of their intended tissue, the pDNA still needs to cross the nuclear envelope and incorporate itself into the cell's genome. Although lipid nanoparticles possess appealing traits like automated assembly and flexible design parameters, there are still significant barriers to LNPs becoming long-term solutions for diseases through gene therapy. But be careful, do not mistake these barriers for impossibilities, for science has always persevered in the face of setbacks. Barriers are meant to be overcome, and crucial discoveries are yet to be discovered. Thank you for taking this journey through life on our plasma membrane.